if you're watching this video, it'll be as usual. You're going to send in the attendance words. You're going to take a screenshot when I say the attendance words. So at the end of the video, as usual, you're going to email a list of attendance words, including the screenshot of when I said the attendance words. And as usual, the last word for attendance, if you want to call it that, will be asking me one question about the material I presented in the lecture. So as you're watching the lectures, keep that in mind. And if anything comes up, you might want to jot it down, and you can use that as your question. But that's it. Anyway, it's about 12 to 13 minutes before we actually get started. So since you're watching a video, you can go ahead and skip ahead about 12 to 13 minutes if you want to. Yep, that's it. All right, it is 8 o'clock. No one is physically here, but we're going to go ahead and get started anyway. I'm glad I see some people online, so I'm not talking to myself. The attendance words will be as usual. I'm going to ask you a question, and that'll be the answer to that will be the attendance words. And also, as usual, the last attendance word, if you want to call it that, will be me saying, or me asking you to ask me one question about the material that I covered today. And again, it's not only does that help you possibly understand something, but it also helps prove to me that you were paying attention to the material. Anyway, let's go ahead and jump into it. I'm going to do the announcements and reminders later. I'm going to wait until some people show up, hopefully. All right, here's where we left off. We left off talking about cytokinesis. Cytokinesis, if you remember, I'll try to draw this. If you think about the entire cell cycle, right, the entire cell cycle is broken down into two parts. First part was interphase. Oh man, it's just disappeared on me. Anyway, interphase was the first part that I tried to draw there. Oh, good grief. Anyway, the second part is the mitotic phase. The mitotic phase itself is broken down into mitosis and cytokinesis, right? And cytokinesis, and they kind of overlap. They are two different things, but they overlap. So that's what we finished talking about, right? So we finished talking about the entire cell cycle. We finished talking about the mitotic phase. And obviously the thing that ends the mitotic phase is cytokinesis. So that's where we left off. And as a reminder, just so you guys know, like some of you missed that question on the pre-lab for this week. And I warned you guys last week, I said, you're gonna get a question like this. Cytokinesis is not part of mitosis, right? So that question that says, name the parts of mitosis in order should have been prophase, metaphase, anaphase, telophase, not cytokinesis. Obviously, that's an important part, and it is part of the mitotic phase, but it's not part of mitosis. So the first word for attendance is cytokinesis a part of mitosis? Yes or no? And to be clear, I'm not saying is cytokinesis part of the mitotic phase. I'm saying is cytokinesis a part of mitosis? Yes or no? That is your first word for attendance. I know you guys are online, but are there any questions before we jump into the new material? All right. So there are some exceptions to the rule. So here we are, we're going back to talking about the cell cycle. And this is what, this is a picture from your textbook. This is what we talked about, right? We talked about how you go from the mitotic phase to G1 to S phase to G2, and then, then you're back to the mitotic phase, right? So there's actually some exceptions to this. Some cells do not follow the classic cell cycle the way I taught it. Now, before I even get started on this, I'll go ahead and let you know there's going to be very minimal questions about this on the exam, if anything. It's important to know that, admittedly, for this 100 level course, this is a little bit complicated, right? So for the exam, I feel like you can just focus on the regular stuff that I taught you about the cell cycle. For the exam, I'm not going to focus too much on this, these exceptions to the rule. But again, I do want you to learn them because to understand how cells work and how the cell cycle works, you need to understand this. And unfortunately, one day, you might need to know this stuff. You or someone you love might come down with some kind of disease, and you might find yourself doing some sort of research on it. And for a lot of stuff, especially cancer, it's going to be important to know how the cell cycle works. So that being said, let's talk about it. We call this the G0 phase. That's what some people might read that as G0, but it's pronounced G0, not that it matters. And in this case, the cells are not actively preparing to divide, right? So you learn in G1, like right from the beginning, which is, G1, cells normally are preparing to divide, right? They're growing, they're accumulating energy, they're accumulating material. But in this case, they are not. This is an inactive stage where they have basically exited the cell cycle. Sometimes this is triggered temporarily by some sort of external signals that you can look into for independent work if you want, like what kind of external triggers signal this. But you don't obviously need to know that for the exam. And sometimes certain cells will remain in the G0 phase permanently, meaning they never do duplicate, right? And then they never do split and form new cells as in mitosis. So again, this is a good independent work topic. And sorry to get off topic here again, but I want to remind you to do independent work, right? Some of you haven't done 0, and you need 100 points by the end of the semester. So let me put it to you this way, and I've said this before, but I'll remind you. If you want to, just write something short and sweet. Ask yourself a question. Just ask a question and answer it. And if that's all you're doing, then you don't even have to do like if you're just literally going to have 2 sentences, right? The question and the answer, then you don't even need to put it in a Microsoft or in a Google document. Just send me an email, and I won't even grade it for length. I'll just give you 1 point for every question you answer, right? So you can say, I was wondering which cells typically remain in the G0 phase. I looked it up, and according to this source, and then you can put in quotation marks and literally just copy and paste the answer, right? So please get some independent work done. It's worth an entire letter grade. Anyway, yes, so that's a good topic for independent work is what kind of cells do this? Or like I said earlier, what causes this? Those are things you can look into. Any questions about this slide before I move forward? All right, so the cell cycle itself, we did say previously, I did say that you need to know for the exam that interphase represents a majority of the cell cycle. The cells are usually in interphase 90% of the time. And you did need to know that, not the number, but the fact that a large majority of the time the cells are spent in interphase as opposed to mitotic phase. But it is variable, right? So it's not like every cell spends exactly 90% of their time in interphase. It's variable. It varies amongst different organisms, right? So humans versus bacteria versus, I don't know, pine trees, whatever, all these different bacteria. It varies by organism, but also it varies within individual organisms. So, you've got all these different types of cells in your body, kidney cells and nerve cells and, all these different cells in your body, and they all vary, right? The length that they're in different parts of the cell cycle is variable. And that's another independent work topic if you want to look into it. Choose a particular type of cell that's in your body and look up, like, how often are they in this phase? Anyway, specifically, or not, this isn't that specific, but a little bit more specifically, within humans, the frequency of the cell cycle length ranges from hours for some cells to a lifetime for other cells. And again, this is something you could look up for independent work. Pick a cell, and how long are they in a single cell cycle, or how long are they in interphase? These are all things you can look up. What is important here, not necessarily for the exam, but to understand some of the things I'm about to teach you, is this last bullet point. The timing of the cell cycle is controlled by internal and external mechanisms. So it's not like your cells are just going freely, right? They're being controlled, and that's what we're about to talk about. So are there any questions about this slide before we dive a little bit deeper into this last bullet point? I heard a noise. If you do have a question, this time you probably raised your hand or you typed something, but if you do have a question, if you could just take yourself off mute for a second and ask the question, that would be great. All right, anyway, so let's dig deeper then. Let's look into this third bullet point a little bit more. Daughter cells are supposed to be exact duplicates of the parent cells. You already know this. This is me saying the same thing over that I've already said. That's definitely going to be a test question. We're still talking about mitosis, not meiosis, right? So you know that mitosis, you start with one cell and you end up with two identical cells, right? Two genetically identical cells. That's what mitosis gives us. But sometimes there are errors, right? Errors specifically in the S phase of interphase, not specifically, but that's the biggest time you would see it, right? When we're duplicating the DNA, a lot of times there's errors in that process, amongst other places. And these mutations, well, it says can be inherited, but you know what? It should say will. Well, yeah, I'll stick with that. These mutations can be inherited by all subsequent cells. In other words, when these mutations happen, if something doesn't fix it, if there isn't some kind of process to fix the mutation or fix the process, then for sure every cell that comes from that mutated cell will have that mutation, right? And we're going to talk a little bit more about this bullet point as well. So because of this, your cells have control mechanisms at these checkpoints. So again, it's not like your cells just are on autopilot going from G1 to S phase to G2 to the mitotic phase, right? There are these checkpoints. And this is what we're going to learn about. These checkpoints are located at the end of G1, right? So it's kind of like before it can leave G1 and go into S phase, there's a checkpoint, and we'll talk about it. Then there's a checkpoint where we leave G2 and go into the mitotic phase, or in other words, the end of interphase, right? So before it goes into the mitotic phase is another checkpoint, and again, we'll talk about that one separately. And then finally, there's a checkpoint during metaphase, right? So remember, metaphase is that part of mitosis where those chromosomes meet in the middle. So these are the three different checkpoints, and we're going to learn about all three of them separately. And the next question, next word for attendance is to tell me how many checkpoints are there in the cell cycle? How many checkpoints are there? We're about to discuss them separately. I've listed them. Now I'm just asking you how many are there. I'm not telling you, or I'm not asking you to name them. I just want to know how many checkpoints are in the cell cycle. That's my question to you. Do you guys have any questions before we move forward and talk about these separately, individually? All right. Let's talk about the first one, the G1 checkpoint. So again, to remind you, this is the end of G1, right? So this is the checkpoint that determines if the cell is going to leave G1 and go into the S phase. And remember, S phase is when the chromosomes duplicate. right? So this is the checkpoint before that. This checkpoint determines if conditions are favorable for division, for cell division to proceed. It is also called the restriction point, because the reason it's called that is because once it goes past the G1 checkpoint, the cell irreversibly commits to cell division. Now, granted, there are other checkpoints that we're going to talk about later, but at that point, obviously once it leaves G1, it is now going into S phase. So it is going to commit to cell division. Even if it doesn't do it, finish the whole process, it's at least committed to starting it, right? It's going to duplicate its chromosomes in the S phase because S comes after G1, right? So this checkpoint, when we said it determines if conditions are favorable, that's kind of like this right here. It checks, what we're doing is checking for adequate reserves. Like, are there enough biological molecules? Is there enough ATP? Whatever it is that your cells need, they need to make sure there's, adequate reserves of them. It checks for the cell size, and it's going to check for damage to DNA. And this, as far as the conversations we're going to have today, this third bullet point is probably the most important, because this is damage to DNA, this mutations that are going to be the most important. And again, I'll remind you, this is mitosis. And in mitosis, right, the end result of mitosis is we have two identical copies of cells, right? So if there's damage to the DNA and it doesn't get noticed in this G1 checkpoint, then it's going to go into the S phase. And that damage to the DNA, if it's a mutation, is going to be copied, right? So then the two cells that result from this process are also going to have that mutation. And then all the cells that they create are going to have that mutation. And all the cells that they create are going to have that mutation, so on and so forth. And that is very important today because we're going to talk about cancer, and that has a lot to do with cancer. And this is also a very important concept later for when we talk about natural selection or in evolution. None of that would happen without mutations. So although mutations are generally bad, they're also very important for life, and we'll cover that later. But yes, this last bullet point is very important, not necessarily for the exam, but for the stuff you're going to learn later today and later in the semester. So again, this is a G1 checkpoint. If these things aren't met, assuming that it's working correctly, then the cell will not go into the S phase, which brings up an independent work question. So what happens, right? If it doesn't meet the requirements, it doesn't go to S phase, is that cell just permanently stuck in G1? Or are there processes, right? So, for example, if there's not adequate reserves, then does the cell just wait, and then once there are adequate reserves, does it then go into the S phase? Cell size, right? So if the cell isn't the right size, does it just wait until it is, and then it goes into the S phase? Damage to DNA, right? What happens if the damage is found to the DNA? Is it just fixed, or does the whole process stop? These are things you can look into for independent work. Before I move forward, I'll go ahead and remind you, for the exam, this is not important. This is a 100 level class. I'm not going to get into this much detail on the exam. But again, this is important for understanding of how cells work, right? For your everyday life, for you to understand cancer, for example, you need to understand this, especially this third bullet point. But again, for the exam, not very important. Any questions about the G1 checkpoint before we talk about the next one? Okay, next we'll talk about the G2 checkpoint. Again, this is the end of G2, right? So this is the thing that's, after G2, we go to the mitotic phase, right? So this G2 checkpoint is kind of like the gatekeeper. It's like, all right, can we leave G2 and go to the mitotic phase? And another way you can think of it too, remember G2 is the last part of interphase. So in a sense, you could also think of it that way. It's like, all right, this is the thing that's keeping us, this is where we leave interphase and go into the mitotic phase, right? And this is the checkpoint to make sure to see if that happens. And yeah, that's basically what this bullet point here says, right? It bars entry into the mitotic phase if the conditions are not met. And the conditions that your textbook says, the conditions that I'm teaching you, are that the cell size and the protein cell reserves have to be adequate, right? So they're assessed and they have to be adequate. We also want to ensure that the chromosomes have replicated properly and have not been damaged. And later in this semester, later in this chapter, hopefully today, we're going to talk about some of this too. Like, so again, mutations, like we mentioned in the last chapter, last slide, we'll talk about some of that. Also, remember when we talked about, well, no, I'll just leave it at that for now. I don't want to jump ahead. But again, for independent work, if you want to look into it, not that you need to know any of this for the exam, but what happens, right? So again, this is checkpoint. And if the cell size isn't accurate, isn't proper, if the protein reserves aren't, what they need to be, if the chromosomes weren't replicated, or if there was some damage to the chromosomes, then what happens? I told you what happens, the basics is it does not leave interphase and go into mitotic phase. But then what happens? Is it just stuck forever in the G2 phase until it dies? Or, does it wait until the cell size is good? Does it wait until the protein reserves are good? Does it fix these chromosome problems, right? If there are some. These are all things you can look at for independent work. And that's it for the G2 checkpoint. Are there any questions about the G2 checkpoint before we talk about the last one, which is the mitotic checkpoint? All right, or the M checkpoint, if you will. This happens obviously during mitosis, specifically at the end of metaphase. If anything, if I asked you any questions about the exam, about this on the exam, that would be it, right? When does the M checkpoint happen? And it happens at the end of metaphase. This is also known as the spindle checkpoint. And what this means is it determines if the sister chromatids are attached to the spindle. So remember, at metaphase, you got all these chromosomes lined up in the middle. So in this case, I'm drawing three chromosomes, right? And then they have these spindles attached to them, because those spindles are the things that move the chromosomes, right? So this is what metaphase looks like, right? Well, for us, it looks different because we have way more than 3 chromosomes. But yeah, you got those 3, you got your chromosomes lined up in the middle, the spindles are attached. And this M checkpoint, it's really looking for this. It's making sure that these spindles are attached. Because if they're not, here, let me draw another one. This organism, this hypothetical organism, only has 4 chromosomes. Now look at here, I'm drawing it. So in this particular case of this horrible drawing that I just made, this chromosome only has 1 spindle attached. So what's gonna happen in anaphase? This is gonna go this way, this is gonna go this way, right? This chromosome, this sister chromatid is gonna go this way, this one's gonna go this way, this sister chromatid is going to go this way. This sister chromatid is going to do that, right? So these 3 chromosomes here, sister chromatids, they're going to do what they're supposed to do. They're going to split up. They're going to split apart, right? In anaphase, that's what happens. They split apart, right? So you no longer have sister chromatids attached to each other. You now have full-blown, full-fledged chromosomes. But in this case, this is where I drew here. Now you're going to have this whole thing. Both of those sister chromatids are going to get pulled that way. So at the end of it, you're going to have 2 cells. One of them is going to have, let's see, 1, 2, 3, 3 chromosomes as it should. And this other one over here is going to have those 3 chromosomes, but it's also going to have an extra chromosome. And not just an extra chromosome, but 2 sister chromatids, right? And so it's going to have those 3 chromosomes, but also it'll have that one. And we're going to talk about that later too. And again, that will be very important. As a matter of fact, the last part of this week's lab deals with this. There's a word for this. It's called non-disjunction. I'll officially introduce you to that term later, but now there you go. So that's what the M checkpoint's for. It's to make sure that these chromosomes are attached to the spindle so we don't have any accidents like the one I just drew here. And again, for independent work, look into it. What happens, right? What happens if the sister chromatids are not all attached like I just drew? Is there some kind of mechanism that fixes it? Does the cell just sit there and wait until it just fixes itself, at which point it moves forward? Or does the cell, is it just done, right? If it doesn't need it, does the cell just say, okay, this is not going to work, so we're just going to be in metaphase until we die, right? Those are things you can look into. And one last thing I want to mention, because I don't think I did mention it before. When we're talking about these checkpoints, G1 checkpoint, G2 checkpoint, N checkpoint, they are not perfect. This is why we have mutations. This is why we have extra chromosomes and not enough chromosomes, right? These things are not perfect. They do result in mistakes. And those mistakes are going to be very important a little bit in this chapter and later in the semester, because again, like for example, natural selection wouldn't happen without mutations. Like we would not be here if there were no mutations. So again, that'll be a very important concept moving forward. That's it for the checkpoints. Does anybody have any questions about the checkpoints? All right. So my next question for the next word for attendance is to answer this question. This is going to be a multiple choice question, by the way. When does the M checkpoint happen? Does it happen in prophase, metaphase, anaphase, or telophase? And this is not a trick question. This is just making sure you're paying attention. And if you're paying even a little bit of attention, this is an easy answer, or an easy question to answer. When does the M checkpoint happen? Prophase, metaphase, anaphase, or telophase? And I'm not even going to get specific, like at the beginning or the end or the middle. I'm just saying which of the 4 phases does the M checkpoint happen? That being said, let's move on to the last bullet point of this chapter. If there are no questions about what we have so far. All right, here we go then. The last part of this chapter. I'll go ahead and tell you now, for the exam, it's not very important. I'm not going to have many questions about this, but think about this. This is probably one of the more important, sadly, one of the more important things you're going to learn this entire semester. Because sadly, you or someone you love is probably going to come up, get some form of cancer. And, you're probably going to want to look into it and learn about that, because every type of cancer is different. So this information is giving you a good background. So when you do have to learn that stuff, you're not starting from scratch. Anyway, before we do that, now that it's 8:23, hopefully there's still nobody here physically, but hopefully some more people have logged in. Let's do the announcements and reminders. So again, I'll remind you that the exam 3 and the exam 2 retake are due the Sunday after spring break, or the Sunday at the end of spring break. And you might think, hey, why are you making me take an exam over spring break? And the answer is, I'm not. The exam is open. You can do this before you go to spring break. But yeah, that's the due date. I'll remind you that it's going to be 50 questions, or about 50 questions, and you're going to have 50 minutes to take it. If you want more time, we can meet online, because if I can ensure that you're taking a closed book exam, I don't care how long it takes you to take the exam. I'll remind you for the retake, which, as far as I know, nobody has done it. It's blowing my mind. Some of you really, really need to do better with your grades, and no one's retaken the second exam. So I'll remind you, for the retake, it's open book. You can retake it as many times as you want. It's not timed. And if for some reason you keep retaking it and you get a lower grade, can't get a higher grade than the first time you took it, then we'll just keep your original higher grade. There's nothing to lose. Also, your first 3 exams are used to write your final exam. So exam 2 is, in a sense, a study guide for your final exam. So for the life of me, I can't understand why nobody's retaking the exam, which will remind me, the last reminder too. If you have a question, if you could just take yourself off mute and ask it. I assume that's what that noise was.
Yeah, is it exam 3 and exam 2, or is it exam 1 retake and exam 2?
Good grief. I'm so sorry. You're right. I'm way ahead of the game. I'm so glad you said that. Matter of fact, whoever that was, when you send in your attendance words, please remind me that you said that. I'll give you some extra credit. Good grief.
Thank you.
Thank you. No, man. Anyway, yeah, exam 2 is due at the end of spring break, and the exam 1 retake. Yeah, so thanks. And my last thing that I want to say about that, and this is where some students forget this, you also have to tell me when you're done with the retake. So if you've retaken the exam, I have no way of knowing. So you have to send me an email and say, hey, I'm done retaking exam 1, and I'm satisfied with the new grade. And then, email me, let me know, and then at that point, I'll go in and change your grade. And to remind you, the grade is going to be your original grade averaged with the retake. So anyway, I'll also remind you that I think it's the Monday. I think when we get back from spring break, midterm grades are due. So just a heads up with that. Again, retaking the exam is going to help. Obviously, you're going to have a better grade when I submit the grades. Also, and I've warned you this before, so I want to do it again. I don't want you to be surprised. After I've submitted independent midterm grades, that's the last time the official grades go into the system until the end of the course, right? So after that, then I'm going to put your independent work grades in. So unless there's like one person finished independent work a while ago, other than her, the rest of you are going to see your grade drop. But keep in mind, it's unofficial, right? Nothing's official. It's just going to be your grade in the grade book in My Online, right? Nothing is official until I resubmit the grades at the end of the semester. But the reason I'm doing this now is because I'd rather have you, I want you to see that shock in the middle of the semester. What I don't want to do is put your independent work grades in after it's due, which is the last day of class. So imagine it's the last day of class and you have a B in the class, but you haven't done any independent work. And then the next day I put in your independent work grades and you now have a C, right? So I don't want to give you that surprise at the end of the semester. Another thing, this is the first time I've told you this, but I'll remind you between now and then. The Monday after Easter, we will be 100% virtual. We're not going to meet in person for lecture or office hours. I plan to go out of town because it's Easter. I'm going to go see my family. So yeah, be prepared to be online. Also, just to let you know, things go wrong. So if my car breaks down and I'm stuck in Kentucky, I mean, probably I could still, get online at a hotel, but just in case, like I said, 100% be prepared to meet online that day. But if something goes wrong and everybody's online and I'm not there, you know why. That just means something went wrong and you don't need to worry about it. If something goes wrong, I'll probably share a video with you of a lecture from a previous semester. And then finally, I'll remind you one more time, let me help you. Please let me help you. So much of your grades and my time and your time would be so much better if you let me help you, right? So when you're working on an assignment, you can come and let me help you get through it. Or like some of you really should have done that with before you submitted the photosynthesis lab. Or again, just let me check your work before you submit it. And you can say, hey, can you just tell me if this will get any credit? Because I can just point out the big mistakes if you want, or I can just thoroughly go through it with you and help you with your grade there. And finally, the third option is after I've graded something, you can meet me online so I can give you detailed feedback. And that is so important that I've been bribing you with a 5% boost to that assignment's grade. So for example, once exam one retake is closed, you can meet me online. I'll give you all the correct answers. We'll talk about all your wrong answers and you'll get a 5% boost to your exam grade. And you'll have all the correct answers so you can help study for the final exam. Anyway, that's it for announcements and reminders. Please, 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 please let me help you. A lot of you really, really need it. And I've just been doing this so long, I already know that some of you who really, really need the help at the end of the semester are going to say, hey, is there anything I can do about my grade? And I'm telling you the answer is no, because I'm telling you now, yes, now there is something you can do about your grade. I'm here to help you. I'm here to teach you. I'm here to boost your grades. But I'm not going to do it, with 1 week left of the semester. So anyway, any questions about the announcements and reminders? And for that matter, do any of you guys have any announcements that you would like to share with the class? All right, let's move on to more fun stuff and talk about cancer. Fun stuff, right? Cancer is a disease of the cell cycle, right? So we just learned about the cell cycle, and that's basically what it boils down to. Cancer is caused, or you could say it is, right? It is a problem of the cell cycle. When things go wrong in the cell cycle, that's what cancer is. And this happens when we get mutations in the proteins that control the cell cycle. So we were talking about the cell cycle, right? We talked about those different checkpoints and these things that allow the cell to go forward, right? Or not go forward. And right, the timing, right? And what I didn't say then, and I'm telling you now, is that there's proteins involved in all this, right? There's all these different proteins that are involved in starting and stopping the cell cycle, right? These proteins that are controlling the cell cycle. So if those proteins have a mutation that prevents them from doing their job, then that's going to result in uncontrolled growth of the cells, right? They're just, they're not going to stop. They're just going to go, 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 go. And that is what cancer is, right? Cancer is basically uncontrolled cell growth due to mutations. specifically mutations in the proteins that control the cell cycle. So are there any questions about this first bullet point before we move to the second one? Okay, so again, because of these mutations in these proteins that control the cell cycle, cancer cells do not respond normally to the control system, right? They divide excessively, and then where it gets really bad is these excessively dividing cells may invade other tissues, right? And that's why for some reason, or for example, that some cancers are more deadly than others. I mean, there's a lot of things that go into it, but yeah, that's one of the reasons. That's another thing I want to say too before I move forward. Keep in mind, this is a 100 level biology course for non-biology majors. So we are really just scratching the surface. I'm giving you the basic foundation, so you can learn more if you need to. But this is very much so an oversimplification of cancer. Anyway, any questions about this slide before we move forward? All right, so like I said, there's proteins that control the cell cycle. And what I've mentioned before, and I haven't mentioned it too much yet, and you're gonna learn more about it later in the semester, the proteins, the instructions on how to build proteins, and I mentioned this before, but here it is again, those are written in your genes, right? So your genes are the codes in your DNA, your A's and your C's, your T's and your G's. Those are your genes, right? So genes, there's instructions for genes on how to build proteins. And for every protein in your body, there's a gene that directs your cell on how to build that protein. So if we're talking about these particular proteins that control the cell cycle, because they are a protein, there's also genes, right? There's genes that are responsible for those proteins. So you might need to know about these for the exam. Probably not, but let's talk about them anyway. Again, this is very important for your everyday life, maybe not so much for the exam. One type of genes are called proto-oncogenes. These are the genes that are responsible for the positive cell cycle regulator. These are just normal genes, right? So these are the things that make the cell cycle go, right? This is the ones that I was talking about, right? Those proteins that are responsible for controlling the cell cycle, that's what these are, proto-oncogenes. Yeah, and for independent work, you could look it up. You could, give me some examples of some proto-oncogenes. Then there's oncogenes. These are mutated versions of the proto-oncogenes. So these are the bad guys that we were talking about in the previous slide, right? These are the ones where, you have the normal proteins like we have here, and then they might have some mutation. And if there's a mutation in the genes, then obviously there's going to be a mutation in the protein that results from those genes. So yes, oncogenes are the mutated versions of the proto-oncogenes. These are the bad guys. These are the things that cause cancer, or that can cause cancer. However, it is worth noting too that they're usually not even harmful. So for example, if there's a mutation in these oncogenes, right? So again, there's proteins that are involved with the cell cycle, proteins that give the start and go signal in the cell cycle. So if the mutation has a permanent stop signal, right? If that's how it breaks, then it's not as bad because then it just stops the cycle, right? So for example, if a cell is in G1, it's in that G1 checkpoint, and it's trying to go to the S phase, right? But there's some kind of mutation in one of the proteins that allows this to happen, then this just doesn't happen. And that cell is then stuck in G1 until it dies, which is bad for that one cell, but that's not that big of a problem, for the rest of you, if we're talking about you as a human, right? But it could be bad if, for example, it increases the cell cycle. So if for some reason it's a mutation that basically gets rid of the stop signal, if you will, right? So now instead of stopping it, it's not stopping it at all. It's just going and going and going, right? So again, big picture here, there's proteins that are involved in the cell cycle, that start it and stop it and all that, they control it. There's genes, obviously, that are responsible for those proteins. Normally, they're called proto-oncogenes. And when they have mutations, they are called oncogenes. Usually, they're harmless because usually they just kill that one cell. And think about it, one cell is not that big of a deal. But sometimes, if the mutation is just right, it can be harmful because rather than stopping the cell cycle, it's like on autopilot and just keeps going. So are there any questions about this slide before we talk about some more genes? All right, the next genes we're going to talk about are tumor suppressor genes. These things code for the negative regulator proteins, meaning the stop, right? There's start signals and go signals, right? These are the stop signals. These proteins can prevent uncontrolled divisions. So again, there's proteins involved in the cell cycle, right? And these particular genes, these tumor suppressor genes, result in these tumor suppressor proteins. As a matter of fact, now that I think about it, no, I know why. I don't want to confuse you. Anyway, so if these genes get mutated, then this is going to result in genes dividing without checkpoints, right? There's no quality control. So all that G1 and G2 and those M, right? Those 3 checkpoints that we talked about, that's what we're talking about. These are the proteins involved in that. And if they get screwed up, that's not going to be good. And here's an example. This is a picture from your book. So here we have this particular gene. This is one of those tumor suppressor genes that happens to be called P53. Not that you need to know this. Normally what happens is if there's DNA damage or there's cell cycle abnormalities or there's hypoxia, which is not that you need to know this, but it's just low levels of oxygen. If this happens, then this tumor suppressor gene or the protein that it codes for, which is P53, it does its work, right? So it stops the cell cycle. It's like, all right, this isn't good. We can't move forward. It stops the cell cycle. And then it can do one of 2 things. It can, and this is one of those things I was asking you about earlier that you can look up for independent work. And I know I'm giving you the answer, but you can still look up some of the other stuff. But anyway, this protein, when it sees that one of these problems happens, it says, all right, let's stop this process, right? That's step 1. And then from there, it either fixes the problem and then restarts the whole thing, or if it can't fix the problem, then it basically kills itself, right? It kills the cell. The cell will kill itself, which is called apoptosis. So that's what happens when you have a normal P53 gene, right? It's a great quality control mechanism. Now, if that gene is mutated, then it doesn't recognize the DNA damage or that there's cell cycle abnormalities or that there's hypoxia. And then it continues. And then that can cause the cells to become cancerous. Yeah, this picture is from your textbook. You can look at it if you want in your textbook. I'm putting a big X through it, indicating I'm not going to ask you any questions about the P53 gene. I probably won't, and remember, or excuse me, the P53 protein. And I probably won't even ask you about tumor suppressor genes in general. But yes, P53, it just happens to be one of those tumor suppressor genes / proteins. Which brings me to another good independent work topic. I just told you P53 is one of those tumor suppressor genes. What are some other ones? You can look at it for independent work. So my next word for attendance is, this is a yes or no answer. Will I ask you about the P53 gene on the exam? Yes or no? Will there be questions about P53 on the exam? Any questions about this last slide for this chapter? All right, we are ahead of the game, and this is the end of the chapter, so I'm going to end it here. This will be the end of the lecture. We're done 10 minutes early. Again, don't forget to send those words in before 9 a.m. Don't forget that you still have to, your last word for attendance, other than that yes or no question I just asked you, is you need to ask me one question based on the material that I just presented. It's to provide, evidence that you paid attention to the lecture. And that's it. I'll be online 9 a.m. at the latest for office hours, and I'll also be in my office. So if you need help or want help, please let me help you. And you guys have a great week.